Hello, my name is Nashat Shabansal, biology teacher from SUZA TV. In this lesson, we will learn about scientific processes in biology. We all have the questions about the world. We ask ourselves many questions from the things we observe and see in our everyday life. For example, why do birds fly? How do millipedes feed? Why butterflies like the flowers, and so on? All these questions can be answered through the scientific process. The scientific process is an organized way of answering a science questions. So the study of biology, like any other science subjects, involves scientific processes. The scientific processes involved in the study of biology include the basic skills of observation, measurements, and experimentation. Through these basic skills, you can answer all the questions about the natural world. Let's start to discuss these skills one by one, starting with observation. Observation refers to the receiving knowledge through what we see, hear, smell, taste, or feel from the outside world. Can you identify parts of human body which enable us to get knowledge through observation? I think this question can be answered by any student. But if not, get close to your television. To see, we use eyes. To hear, we use ears. Smelling, we use nose. And to taste, we use tongue. Skin, on the other hand, used for feeling. Example, feeling hot or cold. Let us now see the function of various sense organs in making observation. Eye. We use our eyes as a sense organ for vision. By using our eyes, we are able to see and differentiate size, colors, and shape of various organisms, and hence we can learn about them. Example, we can identify various biological reagents by observing their color. Example, copper sulfate solution is blue. The Benedict solution is blue color, and Sudan 3 is brown color. In various experiments, color change is used as a criteria to suggest the presence or absence of something. Example, changing of color used to confirm the presence of different food types in a given food sample. Ears. Sometimes you can just use your ears to study various biological concepts. Ears are sense organs for hearing. Many organisms produce different sounds which we can use to identify them. For example, you can differentiate the sound of cats from the sound of cow. Also, how do you know that there is a water inside the coconut? It is just by shaking the coconut and hear the sound of water inside it. Nose. Nose is a sense organ for smelling. In the scientific study, we need to smell in order to identify and distinguish between various things. For example, we can distinguish the smell of ripe mango or oranges from the ripe bananas. We can also distinguish the rose flower and hibiscus flower by their smell. As a scientist, you must be able to use your nose as a sense organ effectively and successfully. But avoid smelling anything in the laboratory without the permission from your teacher or laboratory technician. Skin. We can use the skin as a sense organ to detect heat, temperature, pressure, and even pain. For example, during a hot day you feel hot, while during cold days you feel cold. Even if you close your eyes and someone rubs your skin using a block of ice, you can simply tell it by just feeling the coldness it imparts to the surface of your skin. Also, we can describe and distinguish the texture of many substances by touching them. Example, we can tell how rough and how smooth the things are, such as by touching lemon and orange. You can tell their texture. Also, by touching earthworm and grasshopper, and so on. Tongue. We use tongue to taste various things. By use of tongue, we can differentiate various tastes and be able to discover the type of the taste concerned. For example, one can differentiate salt from sugar solutions by just tasting using the tongue. But avoid tasting anything in the laboratory unless you are told to do so by the teacher or laboratory technician. The second skill is measurement. Scientists use specific instruments and units of measurement in their investigation to determine various quantities such as temperature, mass, length, time, and so on. Measuring is important in collecting, comparing, and interpreting data. It helps the scientists to classify and communicate with others. The standard system of measurement is used all over the world in order to ease the communication of the scientists and to make the data understandable to anyone and in any place in the world. The system is called International System of Units, SI unit. SI unit is a series of units that are accepted and used throughout the scientific world. Using the SI units is to ensure precision and the presentation of the scientific results, international uniformity of the data, and ability to confirm results from other parts of the world. So for the world to see your work, you need to convert them in SI units. Let's see some of the quantities, their instruments, and the SI unit used in scientific process and how to convert them. Starting with measure of length. Length is the distance from one point to another. The SI unit of length is meter. Length can be measured using ruler or tape. Other units for length are millimeters, centimeters, and kilometers. The equivalence is as shown on your screen. Example, convert 20 meters to centimeters. From the equivalence, 1 meter is equal to 100 centimeters. So 20 meters is equal to 20 times 100, equal to 2,000 centimeters. Measure of mass. Mass is quantity of matter in an object. Mass is measured by balances. Example, beam balance, spring balance, electronic balance, etc. The SI unit of mass is kilogram, but also can be expressed in gram, milligram, and tons. For example, see the equivalence on your screen. Example, convert 5 tons into kilogram. So from the equivalence, 1 ton is equal to 1,000 kilograms. Then 5 tons will be equal to 5 times thousands. The answer is 5,000 kilograms. Measure of time. Physical quantity corresponding to phenomena or an event. In most scientific work, we want to know how long an event lasts. Any time standard must answer 2 questions. First, when did it happen? And second, what was its duration? That is measured with devices such as watches and stopwatches. The SI unit of time is seconds, but also can be expressed as minutes and hours and days. See their conversion on your screen. Example, convert 5 minutes to seconds. From the conversion, 1 minute is equal to 60 seconds. So 5 minutes is equal to 5 times 60, which is equal to 300 seconds. Measure of temperature. Temperature is the degree of hotness or coldness. Temperature is measured by means of thermometer. The SI unit of temperature is Kelvin, K. Others are degree Celsius C and degree Fahrenheit F. These units relate by the following formula as shown on your screen. Let's see how we can convert these temperatures, starting with Celsius to Fahrenheit. The temperature in degrees Fahrenheit F is equal to the temperature in degrees Celsius C times 9 over 5 plus 32. So 0 degrees Celsius is equal to 32 degrees Fahrenheit. Let's see another example. Convert 30 degrees Celsius to degree Fahrenheit. The solution is shown on your screen. From the formula, we put 30 to the C, and then 30 times 9 over 5 is 54, plus 32, the answer is 86. So 30 degrees Celsius is equal to 86 degrees Fahrenheit. Now, do in your book as an exercise, convert these degrees Celsius into degrees Fahrenheit. Also convert the degrees Fahrenheit into degrees Celsius by using the same formula. In the formula, you make the degrees Celsius the subject. For example, convert 50 degrees Fahrenheit to degrees Celsius. See the solution. Now do this on your exercise book. Convert these degrees Fahrenheit to degrees Celsius. Now degrees Celsius to Kelvin conversion. Temperature in Kelvin, K, is equal to the temperature in degrees Celsius, C, plus 273.15. See the formula on your screen. Let's see the example. Convert 20 degrees Celsius to Kelvin. Let's do this together. From the formula, K is equal to degrees Celsius plus 273.15. Now put 20 to the C and plus 273. The answer is 293.15. Therefore, 20 degrees Celsius is equal to 293.15 degrees Kelvin. Your exercise. Convert these degrees Celsius temperatures into Kelvin. To convert degrees Kelvin to degrees Celsius, make the Celsius C the subject of your formula. So the formula will change to C is equal to degrees Kelvin, K, minus 273.15. For example, convert 47 degrees Kelvin to degrees Celsius. Therefore, 47 degrees Kelvin is equal to minus 226.15 degrees Celsius. I hope now you have understood how to convert these units. So let's move on with the next basic skill in scientific method, that is experimentation. As you are in the biology laboratory, you will be conducting an experiment. Experimenting puts together all the science processes skills. With the information we gather from our senses, we can make prediction about the natural world. The ability to make accurate prediction revolved on the steps of the scientific method. These include identifying problems, asking questions, formulating hypotheses, experimentation, observation and recording data, interpretation, and conclusion. Let's discuss them step by step. Identifying the problem. This is the first step of the scientific method. The problem is identified by making an observation. The observation can be made from the environment or from the work of the other scientists. Example of the observation is millipedes are mostly found in damp places. Asking question. The second step where the biologists ask a question on what he or she observed. The question can be answered by finding the evidence. Example of the question from our observation is, why millipedes prefer damp places? Formulating a hypothesis. A hypothesis is a suggestion of the answer to the asked questions. So the biologists have to suggest an answer to his or her question. The purpose of hypothesis is to provide a direction to further scientific investigation. From our question, we can suggest that millipedes prefer damp places because they need a lot of moisture. Experimentation. In this step, the biologists perform an experiment to test the hypothesis. This is done by conducting a carefully designed and controlled experiment. The experiment is one of the most important steps in the scientific method, as it is used to prove a hypothesis right or wrong, and to formulate a scientific theories. In order to be accepted as a scientific proof for the theory, an experiment uses variables to test a hypothesis. A variable is any factor or condition that can be controlled or changed. There are three types of variables, which are dependent variable, independent variable, and controlled variable. Dependent variable. This is the factor or the condition observed in an experiment to obtain the results. Independent variable. This is the condition that is changed or manipulated by the scientist. It is not changed by other variables. The independent variable causes the change of the dependent variable. The third type is controlled variable. This is the factor that is not changed throughout the experiment. Example, the light intensity, pressure, and so on. Now, let us test our hypothesis. Millipedes prefer damp. Take two trays, millipedes, damp soil, and dry soil. Set your experiment as follows. In the first tray, one side of a tray, put a dry soil and the other side damp soil. Then put your millipede at the center of the tray. Observe where the millipede will move, to the damp or dry soil. The direction in which the millipedes move is the dependent variable, the factor that we observe. You can change the content of the moisture to the soil by increasing moisture or by decreasing moisture, and continue to observe where the millipede will move. This is the independent variable, the factor that is manipulated to obtain the results, as it causes the change in dependent variable. In second tray, you can keep only dry soil and millipedes, then observe the direction of the millipede. This is the control experiment. Observation and recording data. When you interact with your experiment, you are using your senses to observe. Observation go hand to hand with data recording. Record the changes of the independent variable and the resultant change in the dependent variable. It is necessary to conduct several trials to ensure that the results are the same. Data interpretation or analysis. After finishing the experiment, the researcher then analyzes all the data and uses it to draw a conclusion regarding the strength of the hypothesis. Example from our experiment. We can see that as much as the water content is increased to the damp soil, the more the millipedes move to it. Conclusion. Conclusion is a statement that summarizes what a scientist has learned from an experiment. If the data proves the hypothesis was correct, the question is answered. On the other hand, if the data disprove the hypothesis, the scientific inquiry continues by doing research to form a new hypothesis and then conducting experiment to test it. In our experiment, we can conclude that millipedes prefer damp areas as there is a lot of moisture. So the data proves that hypothesis correct, and the hypothesis is accepted. Let's see the summary of this process as shown on your screen. I hope you have understood how the scientific process is done in scientific studies. Now, as a biologist, you have to follow these steps in your experiments. And finally, you can prove and come with the new ideas and theories in this natural world. Let's stop here for now. Meet me next time to continue with other topics. See you.